Dr. Adam Boyko presenting for us. Adam Boyko is an assistant professor at Biomedical Science at Cornell University College of Veterinary Medicine. His research focuses on genomic investigation of dogs as a model of genetic disease and evolutionary genetics. His work includes understanding the evolution and genetics of village dogs and the Sniffera pariah dogs found in much of the world today. The role of ingredient genetics on canine longevity and health. Dr. Boyko is a graduate of University of Illinois W26 and received a master's in computer science and a PhD in biology from Purdue University before his postdoctoral research with the Department of Biological Statistics and Computational Biology at Cornell University. He served as a research associate in the genetics department of Stanford School of Medicine before beginning his faculty appointment at Cornell in 2011. So I will turn it over to now Dr. Adam.
Thank you. Thank you very much. Really excited to be here on this cold February Friday. I am a canine geneticist, and so I study lots of aspects of dog evolution and dog genomics. And I always like to start by talking about the advantages of studying dogs. And the primary advantage for people interested in evolutionary genetics in studying dogs is that there's a lot of phenotypic signal. So dogs have more phenotypic diversity than any other land mammal. There's almost a 2 order of magnitude difference in adult body size in dogs across different breeds, and of course they vary in many other ways, phenotypically, including coat color, body shape, behaviors, temperament, a whole bunch of different aspects which are interesting to study genetically. More to the point, they get diagnosed with the same diseases that people have. They get treated with many of the same treatments. So they're spontaneously developing diseases that humans also spontaneously develop. But because they have this breed structure, and you have certain diseases that are common in some breeds and not in other breeds, you have this opportunity to really study the genetics underlying these different things and get insights into human analogous conditions. And you can do this in an organism that, lives, 5 to 10 times faster than a human does, and is a little less regulated as well. So you can do studies much more cheaply and easier in dogs than you can do similarly in people. If we look at some of the phenotypic diversity that we see across dog breeds, things that breeders have selected for over the centuries, we see very obvious differences in coat type, whether a dog has a long, short, or short coat, whether they have wire fur or straight fur or curly fur. And it turns out the majority of the coat variation is really just changes in 3 or 4 different genes. And so we can work out the combination of these genes, and we can very easily identify the genes and the genetic variants themselves just by comparing dogs that have certain coats against dogs that have other kinds of coats. So it's the same mutation that's leading to the long coat in nearly every breed that has a long coat. And it's the same mutation in KRT71 that's leading to a curly coat in nearly every breed that has a curly coat. Similarly, if we look at the body size variation, humans obviously vary in body size, and there's genetic factors that underlie that. Human height is highly heritable, although it takes tens of thousands of humans before you start to identify some of the genes that are driving this height variation. And even if you do larger studies with millions of individuals, you're identifying hundreds of genes, but each one of them has only a minuscule effect on the overall trait, and you're explaining less than half of the genetic variation in the trait with these genes that you can identify by GWAS. In dogs, we can do a study with 1,800 dogs across about 100 breeds, and we can very quickly find a dozen and a half different genes that explain the vast majority of difference in body size across adult dogs. So the significance values are much higher despite the smaller sample sizes, and these genes are much more predictive of the adult size of the dog than the genes in humans are for the stature in humans. In addition to these genetic variants that have been positively selected in different lineages for small size or large size or short coats or long coats, we also have the other kind of genetic variation that we see in dogs, and these are for diseases, right? So we have hundreds and hundreds of different breeds of dogs. We've selected these by taking a reduced amount of genetic diversity in the founder population and breeding it true for the phenotypes that we wanted to have, and then in many cases exponentially expanding that population into the breed that we see today, and doing this in some cases with line breeding to accentuate certain phenotypes. And so you have these populations of dogs that have levels of inbreeding and that have undergone exponential growth. So you have this simplified genetic architecture within the breed, but you also have these new variants that have arisen in breeds, or variants that were present in one of the founder individuals and are now at an appreciable frequency, that are being unmasked phenotypically. So you have hundreds and hundreds of typically recessive disease variants that by chance become relatively common in different breeds, and then can be used to study analogous conditions in humans. And these are things like, bleeding disorders, metabolic disorders, all kinds of different things that can go awry, right? It's sort of the Anna Karenina hypothesis of genetic disease, where all happy families are happy in exactly the same way, but every sad family is sad in its own unique way. And it's the same way we see different disease alleles arising in different breeds of dogs, that then, because of a popular sire, has increased the frequency of that allele, and then back crossing then unmasks the recessive allele. We have these populations now where we can study what these variants do, what they look like, what they are, and see whether analogous genes in people have it, whether we have compensatory mutations that maybe can be protective of it, or whether gene therapies might be useful in addressing the condition. So we've mapped more Mendelian disease variants in dogs than any of these other species in OMIA. And we've also, mapped the variants underlying body size and fur type and body shape and things that have been selected for across different breeds. At the same time, we've sort of lagged in trying to identify variants that underlie complex diseases in dogs, things that are shared across many breeds, like cancer and allergies and other autoimmune diseases or hip dysplasia. We've also found slower progress in understanding the genes that influence dog behaviors. So different breeds of dogs tend to exhibit different behaviors, but it's not quite as standardized as other kinds of phenotypic diversity that we see in dogs. It does seem to be easier to identify genetic variants underlying it. We've also made slower progress in understanding the role of genetics on longevity. So we know that big dogs tend to not live as long as small dogs do. They senesce more quickly on average. But we don't know whether it's just size per se, or whether there's particular genetic variants that are driving that, and whether there's other genetic variants that maybe are protective and can increase the health span of dogs. So with this context, I want to introduce a study that's kind of important to me and many other researchers in the field. This is a study that's been supported by the Morris Animal Foundation, and it's a longitudinal study in golden retrievers called the Golden Retriever Lifetime Study. So this is a $30 million plus study where 3,000 golden retriever puppies were recruited about 12 years ago, and then they were studied throughout their lifetime with annual vet visits. Samples were collected at these vet visits. As these dogs get diagnosed with diseases, it gets recorded. Golden retrievers, of course, are one of the breeds with the highest levels of cancer propensity. About 60% of golden retrievers wind up dying of cancer. We've reached the study milestone of over 500 cancer deaths in these dogs as of 2.5 years ago. And so we have this large data set of golden retrievers that have been studied genetically and through their biospecimens of all different kinds of biospecimens, blood, serum, necropsies of the dogs that have died, nails, hair, urine, and all of these things are stored. So there's over 1 million biospecimens that are stored. This is a project that Morris Animal Foundation really wants scientists to utilize these resources and these biospecimens. So I invite anybody who's interested to apply for a grant through the Morris Animal Foundation website. So they'll pay for you to look at the samples and gather insights from these samples. And you can also go to the data commons. So this is freely available to academic researchers, and you can look at all of the diagnoses that are in the database so far. You can download raw data, including genotype data. They're going to get some sequence data up there soon, all the laboratory results, different diagnoses, and different surveys that the owners have filled out. So this is all available on the Morris Animal Foundation Data Commons. And so using these data commons, been working with a researcher at Cornell, Jess Haywood, and a veterinary student, Reed Milner. And we've been trying to see whether we can take the data that's been recorded at these health checkups and from the genetic information to see if we can get some insights into, I mean, the study is still ongoing right now, but we have this large cohort of golden retrievers that have died of natural causes, and we have this other cohort of golden retrievers that are still alive. And so are there any genetic differences between those two cohorts? Are there any phenotypic differences that we see between those two cohorts? What do we see? What is the influence on survivorship that we can discern, right now? And so looking through all of the data that's been recorded, we have nearly 1,400 dogs that have recorded deaths, which we categorize as natural deaths. So there was about 50 or so dogs where the recorded death was something like, hit by a car, died in a fire, euthanized for aggression, things that clearly weren't related to the health and longevity of the dog itself. But for the rest of them, we categorized it as natural deaths, and we're going to compare that cohort of 1,377 dogs against the 919 dogs where we know they've survived at least 8 years into the study. We don't have any recorded death right now from those dogs. So doing the simplest kind of genetic analysis you could possibly do, where we just take case control. Is it one of the 1,377 dogs that died of natural causes, or is it one of the 919 dogs that has survived? And we already get a marginally significant association on chromosome 14. It's a pretty large locus, nearly 3 megabases long. It's got a lot of genes in it, but many of the genes seem to make some sense for why it might be associated with aging. So this is an opportunity maybe to explore in other breeds and in other cohorts to see if we detect the signal in other breeds and can identify the genetic variant or variants that's underlying it. Now, of course, this is a simplification. There's other things that affect longevity. 2 things that are potentially important are body size and inbreeding. And so we took the body size as a covariate, so the adult body size that we measured in these golden retrievers. And if we look at that, we see for the dogs that died of natural causes, they actually are larger than the dogs that are still alive after 8 years. So even though golden retrievers tend to be similar size, there's a breed standard size for golden retrievers, we still see a significant effect of body size in the same direction that we see across breeds. Bigger dogs seem to be more likely to die sooner than smaller dogs do. In addition, we see a significant impact of inbreeding. So the dogs that died of natural causes on average have an inbreeding coefficient above 29%, whereas the dogs that are still alive after 8 years have an inbreeding coefficient of less than 28%. In both cases of body size and inbreeding, these are not huge absolute differences, but with the sample size that we have, these are significant differences. So there is, based on the normal range of inbreeding that we're seeing in the dogs and based on the normal range of body size that we're seeing in these dogs, statistically significant effects in the direction that we would expect of bigger, more inbred dogs being more likely to die sooner in the study than dying later. There's kind of an interesting bimodal distribution to the inbreeding coefficients that we're seeing in the study. And I think this might point to a little bit of biased sample recruitment. So, owners for these studies tended to be located near research hospitals. They tended to respond to advertisements to join the study. So, they tended to have dogs that, maybe cost a little bit more money to procure. If we compare them to a more normal set of golden retrievers across the United States collected from DNA testing by Embark, we actually see there's a difference. And so these golden retrievers in this golden retriever lifetime study, on average, are a bit more inbred than what we're seeing in a larger cohort of golden retrievers across the United States that had samples submitted for DNA testing. So that's one potential bias that we're seeing in the study is higher than average inbreeding coefficients in these golden retrievers. With that said, taking these 2 covariates of body size and inbreeding, it actually doesn't make a significant effect. We still see the same level of significance, actually slightly more significant for this chromosome 14 locus that we saw before for dogs dying of natural causes versus controls. We can separate this out into the different diseases the dogs were diagnosed with, and we can see that cancer overall is associated with the same locus, which makes sense because cancer was the primary driver of mortality so far in this cohort. The most common cancer, hemangiosarcoma, also seems to have a significant signal at this chromosome 14 locus. But importantly, other kinds of cancer, like histiocytic sarcoma, we're seeing different signals, particularly here on chromosome 5. And this was actually exciting to me because this is a signal that has been shown before in a different cohort of retrievers from a different breed by a colleague, Jacqueline Evans. And so whenever we have complex diseases and we're starting to see the same quantitative trait loci popping up in multiple studies using different animals, it certainly makes me feel a lot more satisfied in the result that we actually have something real genetically going on here, and now we have multiple cohorts we can use to try to identify causal variants. And this chromosome 5 locus, it's not just for the histiolytic sarcoma, which we already knew, but it also seems to be what's associated with lymphoma in this golden retriever cohort as well. So I think we have different cancer drivers at different loci driving different cancers, including for soft tissue carcinoma. We see a suggestive signal around chromosome 19 as a potential driver for that cancer. Now, in addition to the genetic data that was collected, there was a lot of phenotype data that was collected from these retrievers, and in particular, what Jess and Reed and I were most interested in are these clinical blood chemistry panels and complete blood counts that were collected at wellness visits annually for these dogs in the study. And so we have this really clean data set of multiple observations over time of all of these different blood values that the dogs had. And so we're excluding the puppy values because they tended to be a little bit more variable. But we have about 40 lab values that were collected and analyzed each year at Antech for the first 7 years of the study. And we can ask, are any of these early life phenotypes potentially correlated with later survivorship in the study? So we have the blood phenotypes, we have body weights, we do a cleaning of this process, so we make sure that there's not an age effect on any of these phenotypes. We remove outliers, we do normalization, we do sex correction and age correction if necessary. And so then we wind up with a model where we're trying to see whether natural death is predicted by the blood value, including inbreeding, body weight, and sex, spay-neuter status as potential covariates. So the distribution of these blood phenotypes is kind of as you expect. They tend to be normally distributed. Antech publishes reference intervals, and for most of the blood phenotypes, nearly all of these healthy golden retrievers are within the reference intervals. Potentially, there's a little bit of low phosphorus maybe in these golden retrievers compared to what the reference interval is. So that's a potential area to look at later. But then we can see now, do any of these blood phenotypes seem to be predictive of survivorship later in the cohort? And surprisingly, for 3 of them, the answer was yes. So we see this effect of high amylase leading to higher rates of death later on in the cohort, similar to less effect with phosphorus. And then magnesium has the opposite effect. High levels of magnesium seem to be protective and make it more likely the animal is going to be in the survivor cohort versus the early death cohort. And we can sort of trace these out by deciles, so we can see the effects of amylase is really driven not only by these highest decile of amylase having the highest rates of death, but it's sort of this overall linear kind of effect that we're seeing in the cohort, sort of similar for what we're seeing with phosphate, not quite as extreme of a odds ratio, and then in the opposite direction here for magnesium. And it was exciting, particularly to see amylase as one of these associated with longevity later on, because we've already worked out the genetics of amylase quite a bit in dogs. So dogs, unlike wolves, have this copy number variant for pancreatic amylase, AMY2B, on chromosome 6. And so we can see across the world, dogs tend to have very high copy, 10x or more diploid count copy number of amylase, although Arctic dogs and dingoes don't seem to show the same degree degree of elevation, and this seems to track with starch diet. So the amount of starch in the dog's diet increases the copy number for amylase. Having a higher copy number increases the expression and increases the amylase activity in the dog. So this was all worked out, about a decade ago, and more recent ancient DNA studies have sort of gone through, and they've shown that this copy number count has really increased in Europe, between 6,000 and 8,000 years ago, sort of during the Bronze Age agricultural expansion. And so now, these European-derived dogs all have relatively high amylase copy counts compared to other canids and to wolves. And it's sort of an example of convergent evolution to some degree, because we see a similar pattern in humans, and particularly the expansion of amylase genes, particularly amylase 1, the salivary amylase in human populations, again in Bronze Age Europe being one of the biggest periods of these expansions. So these are all complicated copy number variants in both humans and dogs. And we don't directly assay the copy number variant with the genotyping array that we're using, but clearly there's markers on that array that are linked to copy number variation that is important in golden retrievers and other breeds. And so, in fact, earlier on, I had a student, Michelle White, who looked at some of the biobank dogs of various breeds that we had here at Cornell, and we could see this chromosome 6 signal of amylase coming out in our biobank dogs. So we know that the amylase values from the blood tests are correlated with the amylase genotypes that we're seeing here on chromosome 6. And with the cleaner and larger data set from the golden retriever lifetime study, we see a much more significant peak, and we can really localize what's driving this peak at 10 to the minus 270 first power. And we can look at how golden retrievers that carry that carry different genotypes at this chromosome 6 locus actually have different reference intervals for what the typical amylase value is. So if you look at wild type golden retrievers, they tend to be on the lower side of the reference interval. Heterozygous golden retrievers are in the middle, and then you have these mutant homozygous golden retrievers, only about 6% of the golden retrievers, but they have much higher values of amylase than these other golden retrievers do. And if you look at the survivorship in these cohorts, we see the lower amylase, the wild type golden retrievers have significantly higher survivorship than the heterozygous or the mutant ones, which kind of agrees with what we see. So having a higher amylase phenotype seems to predispose for early death, and we see that also shining through in this amylase QTL variant that's associated with it. Now, there's one other blood phenotype that my lab had studied with the biobanked dogs at Cornell, where we saw a significant genetic effect. And this one is a little bit more interesting in some ways because it's alanine aminotransferase, which is harder to figure out why there would be a strong QTL driving this signal. This is a signature of liver damage. So elevated values of ALT are typically a sign of liver damage. That's why this is part of the blood test that's done for dogs or humans. When we look in the golden retriever data set, we once again validate the association that we saw. We get a much stronger association here on chromosome 13. It's right on the GPT gene, which encodes the transferase. So it's got a really good candidate gene for what's driving this. And again, what we see is that depending on the golden retriever's genotype, the reference interval for the golden retriever is going to be very different. And so these homozygous mutant dogs have much lower amylase values than is typical, and they also have a lower survivorship compared to the dogs that are wild type. This lower value for these mutant dogs is actually pretty important, because what we see in the Cornell data, looking across all breeds, is that sure, in the healthy dogs you have a lower mean ALT for these mutant dogs compared to the wild type dogs. But when you have a dog with liver damage, you're going to elevate the ALT values. But for these mutant dogs, the mean of the distribution is 181, so that's above the reference interval. But you see many of the dogs, even though they're elevated, they're still below this top of the reference range of 100. And so you have this much greater potential to miss liver damage if you're just using ALT values as sort of your indicator of liver damage in a dog that has at least one copy and especially two copies of this mutant QTL. And so this kind of points to maybe we need personalized reference intervals for some of these blood phenotypes where we see strong genetic effects. Now, with the 40 different blood phenotypes, we actually could look at other phenotypes as well to see if in the golden retriever cohort we could detect genetic associations that weren't detected earlier in the Cornell Biobank study. And the answer was, pretty surprisingly, yes. Many of these phenotypes had really strong effect QTLs. And in many places, it was a single QTL, relatively common in golden retrievers, and right above an interesting candidate gene for the phenotype. So for instance, in alkaline phosphatase, we see this strong p-value on chromosome 2 right at the alkaline phosphatase gene. Right, so it makes a lot of sense that a genetic variant in this region could cause large changes in the blood phenotype. We see a similar signal with bilirubin. So total bilirubin seems to have a strong genetic association on chromosome 1. And if we look at the genes in this region, it's right above the biliveridin reductase B gene. So potentially variants here are associated with the ability to remove bilirubin from the blood. Creatine kinase has a signature on chromosome 8, right above the creatine kinase B gene. So again, we have a really good candidate here. And triglycerides has a strong association on chromosome 5. And in this case, it's above several different genes that are important for triglycerides, most importantly the apolipoprotein C3, which we think is probably driving this variant, although we don't see any coding variants in the region that seem to be driving it. So we think much of what's going on here is regulatory in nature, and so it'll be interesting to do follow-up studies to see if we can identify any of these putative regulatory variants that may be driving the phenotype. So about half of all the lab values that we saw for these blood phenotypes had significant genetic associations in golden retrievers, and 13 of them had what I would call highly significant associations, where we're talking about p-values of less than 10 to the minus 11th, with the remainder having significant but not as highly significant p-values. We had the strongest signal actually was from complete blood count traits. So there was a QTL on chromosome 9 that was highly associated with eosinophil, monocyte, and percent eosinophil and percent monocytes. And this was the strongest association that we had in the study, and it was right on chromosome 9 at the eosinophil peroxidase gene. So p-value of 10 to the minus 310th. And this was really interesting. It had a big phenotypic consequence. So if you look at wild type dogs in this region, their ratio of monocytes to eosinophils is about 2 to 3. Whereas if you look at the mutant dogs, they have about the same white blood cell count, but now the ratio of monocytes to eosinophils is 8 or 9 to 1. So it's a much different population of eosinophils and monocytes, relative proportions that you're seeing in dogs depending on the genotype that they carry at this chromosome 9 locus. And to us this is interesting because in human studies eosinophil count has been associated as protective from certain cancers. And so you can kind of start to paint a story that maybe the reason why we see so much cancer in golden retrievers is that many golden retrievers have this mutation that leads to much lower eosinophil counts and much reduced ability to fight precancerous cells. Now, we're actually not seeing evidence of that. We don't see this QTLs being significantly associated with survivorship, but it's still a pretty profound difference in blood phenotypes, and it'd be interesting to see what downstream effects that might have. So overall, if you look at human blood phenotypes, there is genetic variation, there is heritability of many of these phenotypes, but the genetic variants that are driving it for the most part are either have very small effect sizes, or if they have a large effect size, they're pretty rare in the population. And in these golden retrievers, it's a very different study. We see many common genetic variants have really profound effects on the phenotype. And in the case of, amylase 2B, sure, we have a story about selection and why it might have gotten selected for, although it's difficult to understand why it might vary so much within golden retrievers. But for these other genes, it's a little bit harder to understand, what's driving this and how do we have common variants that have effect sizes of half a standard deviation or even a standard deviation or more, do they affect downstream phenotypes of interest at all? So this, simplified genetic architecture is, to me, something we expect in something like body size across breeds, where you've had this really strong selection going on. It was more surprising to see it within breeds for blood phenotypes. We could also look at body size within the golden retriever cohort. And now there's not as much phenotypic signal because golden retrievers tend to be the same size, but we do see two of the size loci that seem to be segregating within the golden retrievers, IGF-1 and L-Coral, that seem to be associated with body size. And we come up with a novel suggestive association around this short stature homeobox 2 gene. So I think that there's actually other genetic variants associated with body size that we can find by doing these large-scale studies within certain breeds to identify breed-specific variants that are associated with size, in addition to still segregating variants that we know affect body size across breeds. Now, we know that overall, the phenotype of body size was associated with survivorship, but we can look and see whether the large size allele with IGF-1 was also associated with survivorship, or the large size allele with L-Coral was also associated with body size, or whether there's a difference between these two. And this is kind of an example of Mendelian randomization, where we can see whether genetic effects that change an intermediate phenotype are then also associated with downscale things like survivorship. And so here we're doing association studies where we're including the QTL along with the value to see if it has a significant effect. In this case, when we do these kinds of analysis, so inbreeding is significantly increases probability of death. Intact females have a higher probability of death. The GGT locus seems to increase the probability of death, whereas the ALT locus seems to be protective of death. Interestingly, L-Coral seems to be significantly associated with survivorship, but IGF-1 doesn't. So maybe this is suggestive that different size genes may impact longevity differently. So it'll be interesting to try to replicate this in other breeds where IGF-1 and or L-Coral are segregating to see if we're getting a consistent pattern here or whether genetic background seems to be playing a role. And then we saw this really interesting signal in triglycerides. So in human studies, having high levels of triglycerides is associated with higher rates of cardiac death and also increases in body weight and things like that. And it's, a bit of a question of whether it's the genetic variants underlying triglycerides that seem to be driving the association, or whether triglycerides is just an indicator of a bad diet and other aspects of the diet are what's leading to the cardiac and early death phenotypes. Here we have a triglyceride association where the QTL for increased triglycerides actually seems to be protective of early death, which was sort of in the opposite direction of what we were expecting. And when we look at this further, across different sexes and across different ages, we see that it has the effect of body weight that we would expect. So dogs with the high triglyceride QTL are heavier than wild type dogs, and this is true, regardless of age or sex status, even though the trajectories are a little bit different and the averages are a bit different for different breeds. But these high QTL dogs also have greater survivorship. So even though big dogs, even within golden retrievers, tend to die sooner than smaller dogs do based on body weight, this triglyceride locus that drives higher body weight seems to be protective for survivorship. So it's really an interesting question of what's going on here, and are there nutritional interactions or other things that we can try to tease apart to figure out what's going on. So to conclude, we did find a suggestive association on chromosome 14 that seems to be correlated with survivorship in this cohort. We also see low levels of inbreeding and smaller stature increase survivorship in this cohort, as well as across breeds. We see early blood phenotypes, some of which are predictive of later survivorship. Many of these have QTLs, which offer us an opportunity to study the impact of genetic variants that change that phenotype and do these kind of Mendelian randomization experiments to try to disentangle causality. I think that there's lots of upcoming work that needs to be done. We would love to look at these QTLs across diverse breeds to see if they're common across breeds, or they tend to be more golden retriever specific or retriever specific, or what. When we look across different breeds, we can generally narrow down the association intervals. That helps us identify causal variants. Would love to be able to compare blood work from sick dogs that have been genotyped at these QTLs to see, whether we can define more accurate reference ranges that are going to give clinicians more accurate guidelines about when, based on a dog's genotype, a value is unusual or not. And we would love to, replicate the study in other breeds to see, these specific variants that impact phenotypes like body size or blood phenotypes. Do they affect longevity in consistent ways? And if so, what can we do then to increase the health span of dogs and maybe even people? So I'd like to acknowledge my collaborators at Cornell and elsewhere with this. I am on the board of Morse Animal Foundation, so that's part of my enthusiasm, but it really is a unique and great data set that I encourage everybody to kick around who has any kind of interest in doing studies like this. And with that, I'm open for questions.
Thank you very much, Dr. Boyko, for your presentation. I would now like to open the floor to any questions, either from people here in the room or from those joining us online. We do have a question in the chat. Let me just here. Can you see the question in the chat, or do you want me to read for you?
Yeah, so that is a great question about the runs of homozygosity peaks. Homozygosity mapping actually is a pretty powerful way of identifying regions of association. It's particularly useful when you're dealing with recessive phenotypes, because the cases are all going to share a run of homozygosity. In this case, the QTLs all seem to be additive, and so we don't really expect to have run a homozygosity mapping being that informative. But again, even for additive traits, when you're looking across breeds, if you've got something that's been selected for in one breed and fixed, then you would expect to see that ROH overlap for that breed, but just not in the other breeds.
Great, thank you so much. We do have a question here in the room. Sorry. You can go now.
I was interested about small breeds. So you mentioned large breeds, particularly highly selected large breeds. Longevity. But what about the very small breeds? Do you see the same thing happening?
So I think if we look at the typical breed ranges, I think the smallest breeds are things like Chihuahuas and Yorkies, and those are going to typically be, I don't know, 3 to 5 kilograms, maybe even slightly less. But I don't have offhand exactly what the range is, and those are breeds that survive on average for 15 or 16 years, right? So I think you have this pretty clear decreasing longevity, increasing senescence rate with typical body size. Now, if you were to take a Chihuahua and try to breed it to be as small as possible, and I think the world's smallest adult Chihuahua is, less than half a kilo. I do think you run into problems, and whether that's because you're inbreeding that dog to get the small size, or whether it's just such an extreme phenotype it's not conducive. I mean, I think you see a similar thing in rodent studies where larger mice don't live as long as smaller mice do of the same species, but if you try to really get a super small mouse, it's also not going to live that long.
So partially it could be inbreeding, as you've suggested, when you really highly select for size. But what about physiological aspects, like historically, or like, ancient breeds of dogs would have probably been moderate size. And so would they have an advantage physiologically over the very large or very small breeds?
Yeah, I think there's typically a disadvantage when you breed for an extreme phenotype, and some phenotypes being more disadvantageous than other phenotypes. And we've had small toy dogs for millennia, and we've had large, fighting and livestock guardian dogs for millennia. But there are certainly, with modern pedigreed breeding, more enforcement and maybe more selection for extreme phenotypes in some cases, even including size. I don't know how the world records for the largest and smallest dogs have trended over time, but it wouldn't surprise me if those became more extreme over time.
Thank you.
Please, Samula, you can go ahead.
I was thinking, first, thank you for the presentation. It's pretty amazing. I have a question. I was thinking more about the application of your results. How does I assume breeders or associations, they take this information that you may have some of the dogs that may have markers of mutation that will give a specific phenotype that we don't want. And if there is any efforts to, I don't know, perhaps breed to remove those animals or not breed them in the future?
Yeah, so so one of the reasons that I started Embark Veterinary is I wanted to give owners and breeders kind of the information about their dogs so that they could make intelligent decisions about care and breeding. And one of the things that's offered in that test is what the ALT phenotype or genotype for the dog is, because we think it's important, if your dog naturally has low ALT, that you and your vet are aware of that when you're interpreting blood values, and maybe you want to get a baseline blood value for your dog that you can use to compare later on, and have more resolution for if there's something wrong with your dog or not. We obviously we scan for all of the Mendelian conditions that we can, and we want to give breeders that information, particularly so that they can make sure they make breeding decisions that aren't going to lead to affected animals. It gets a little complicated in that many breeders use the test results and then try to breed out any genetic defects at all from their breeding pool. But of course, the vast majority of these genetic defects are recessive. And if you have a common recessive defect in a breed and you stop breeding any of the carriers, you actually are losing genetic diversity in that breed, because you're not just refusing to pass on that recessive allele, you're also losing all the other genetic diversity that's contained within these carriers, right? And the primary the primary challenge that the breed is going to have over time is maintaining genetic diversity, right? So we encourage breeders to use genetic testing, but use it to make sure you don't breed two carriers together, but not to remove all of the carriers from the breeding pool, because we don't want to introduce this loss of genetic diversity. Because right now, if it's something that you can test for, then you just do the testing and you're never going to create another affected animal, right? If you put a bottleneck on the breed by not breeding any carriers, then there's other stuff that you can't test for right now, because we haven't done the study yet, that's going to get to a higher frequency, and now you're going to have new genetic problems that maybe are going to be more difficult to manage, and you're going to have less genetic diversity in your background.
Yeah, no, I got it. Yeah, interesting. Yeah, thank you.
Thank you. Any other questions? We do have a question in the room.
Hi, thank you for a great presentation. I was wondering if there's been any similar on mixed breeds, maybe weighting by breed composition. Sorry.
Yeah, so there have been studies done, just from clinical data. So like on average, mixed breed dogs live about a year or two longer than purebred dogs at the same size. And there was some debate, is that being driven by inbreeding, or is it that the breeds have their own health problems or maybe extreme phenotypes that are causing health problems or something like that. When we dig into the embark data, we pretty clearly see that the even for both mixed breed dogs and purebred dogs, inbred dogs don't live as long as outbred dogs do. It's just for the mixed breed dogs, more of them are outbred, and for the purebred dogs, more of them are inbred. That's why you see that overall difference. But the trend is the same for mixed breed dogs and purebred dogs, where you get decreasing longevity with increasing inbreeding. And not only decreasing longevity, you see decreasing litter sizes with increasing breeding. You see increased reports of health problems with age, with higher levels of inbreeding. And you see decreased stature. So you don't grow as big as you would have otherwise if you're inbred, if you're a dog. So it's all basically any measure that you can look at for inbreeding depression. We seem to see evidence of that occurring naturally in purebred dogs and in mixed breed dogs that happen to be inbred because of backyard breeding or something like that.
Okay, cool. Thank you. And also, I was wondering what the density of this panel was for this.
Yeah, so for the golden retriever lifetime study, it's a 650K chip with about 500K of the markers segregating in golden retrievers.
Okay, cool. Thank you so much.
Any other questions? No? My question might be, you might have missed that, but I was wondering if there's any particular reason that you have chosen golden retrievers.
Yeah, so I mean, I didn't choose golden retrievers. Rod Page and the Scientific Advisory Board back in 2010 chose golden retrievers, but it was because we really wanted to know what drives cancer in dogs, and then secondarily, what other aspects of longevity can we ascertain? And it's still an open question. Like, are there genetic variants for different cancers in golden retrievers that are segregating, that are driving increased cancer risk for some goldens versus others, or is there some genetic region that's fixed in golden retrievers that's predisposing them all to cancer, and some of them get lucky and don't get it, but lots of them do get it? And so it was really interesting to see these within golden retriever significant association signals suggesting we do have stuff that is segregating within goldens that's associated with longevity and cancer risk. But it'll be interesting to look at crossbreed studies too, to see if any regions of reduced diversity in the golden retriever genome seem to also be predisposing for cancer.
Okay, great. If there's any other questions, I would like to end the meeting for here. And this has been a great discussion. Thank you again for presenting for us today, Dr. Adam. And thank you everyone for coming. The recording will be posted on YouTube, and I will share more details about that with you later. So
Great.
for now,
thanks so much. I appreciate all the questions. Have a good weekend.
No problem. Have a nice weekend. Bye-bye.